Hello everyone, my name is Rahul. In the previous video, we had discussed about endoplasmic reticulum. In this video, we are going to discuss about the next component of endomembrane system, that is Golgi apparatus. Golgi apparatus was first discovered by a scientist known as Camillo Golgi in the year 1898. In plants, Golgi apparatus is known as dictyosomes because they are not present as a complex. So in plants, Golgi apparatus is not present in the form of a complex, rather each particle is present isolated in the cytoplasm of the cell. That is why they are known as dictyosomes. Now moving on to the structure of Golgi apparatus. Golgi complex, it is made up of three components. The first component is cisternae. These are flat, disc-shaped sac-like structures, and many cisternae are arranged in a stack, which are arranged in a stack parallel to each other. The diameter varies from 0.5 micrometer to 1 micrometer. So the arrangement of cisternae is such that they are arranged in this manner parallel to each other. There are varied numbers. Some will have more cisternae, some will have less cisternae. They are concentrically arranged near the nucleus. So if we are asked to find out the Golgi apparatus or to locate the Golgi apparatus, the best place to look at will be near the nucleus. The cisternae have two phases. These cisternae, they have two phases. One is a convex phase. Which is towards the nucleus, or endoplasmic reticulum. This phase is known as the cis phase or the forming phase. The other phase is a concave phase. This faces away from the nucleus or the endoplasmic reticulum. This phase is known as the trans phase or the maturing phase. The second component of Golgi apparatus is the presence of tubules. These are branched irregular tube-like structures. And the third component is vesicles. All three components are arranged as a complex, and they cannot be functional if isolated. So if you separate these components from one another, they will not be able to function well. So this phase, which is the more convex phase, this is the cis phase, and this phase, which is more of a concave phase, this is the trans phase. From the cis phase, the vesicles from the nucleus or the endoplasmic reticulum are going to enter inside the Golgi apparatus, and from the trans phase, these vesicles are being formed to release the substances whether intracellularly or outside the cell. So trans phase can be identified by the presence of a number of discharging vesicles, whereas cis phase can be identified as the convex phase where vesicles are entering inside the Golgi apparatus. Now moving on to the functions of Golgi complex or the functions of Golgi apparatus. The principal function is that Golgi apparatus performs the function of packaging, materials to be delivered either intracellularly or outside the cell. Delivery of substances outside the cell, it is known as secretion. So there are three steps involved in secretion. The first step is Golgi. Golgi receives the materials from the endoplasmic reticulum through its cis phase. These materials are chemically modified these materials are chemically modified. Example: glycosylation and glycosidation of proteins and lipids, thus forming glycoproteins, glycolipids, etc. So all the materials that have been synthesized by the endoplasmic reticulum are modified inside the Golgi apparatus. After chemical modification, these substances are packed into vesicles. These vesicles pinch off from the trans phase and are delivered outside the cell or either inside the cell. So all the macromolecules which are sent to be outside the cell move through the Golgi body. So Golgi is called as middleman of cell or director of macromolecular traffic. The next function is formation of lysosomes. Along with endoplasmic reticulum. The other function is synthesis and repair of biomembranes along with endoplasmic reticulum, synthesis of cell wall material. It is also involved in cell plate formation during cell formation. Golgi changes into the acrosome of sperm upon sperm maturation. So the acrosome is derived from the Golgi apparatus. With this, we finish off with our discussion on Golgi apparatus.